Thanks, Linda, for having me, and thanks to all of you for coming and giving me an opportunity to talk about our work at the laboratory. As you know, the Bioscience Division is actually it's a small component of the laboratory, but we are a dedicated group, and many of us are focused on bringing renewable fuels to the marketplace. And what I'd like to do is really try and give you some, break the ice a little bit at the beginning, and then talk about some basic algal biology and evolution. It kind of give you an algal biology 101 lesson, and then present the case for algae, why we think algae is potentially a good source as a renewable fuel feedstock. And then we're going to dig much deeper into some of our current work, or the work of many of my colleagues, and talk about a couple of case studies with 3 different species of algae that we're currently studying. All right, so let's start from the beginning. So so what really are photosynthetic algae? So there are many different types of algae. Some are photosynthetic, some are not. But the ones of interest to us are the ones that actually conduct photosynthesis. So just like I said, we're going to back up here. So photosynthesis, for those of you who aren't familiar, you take energy from the sunlight and you use that energy to capture carbon dioxide from the atmosphere and convert those carbon dioxide molecules into longer chain carbon molecules. And from that carbon dioxide, these particular algae can form all of the biomass in their cell. You add a few nutrients, you throw them in water, and out comes organic material, and then of course they evolve oxygen. The pictures at the bottom show images of several different types of algae. So algae are multicellular and unicellular. So they have single cells and they can form very complex structures. So this is a type of green algae, this is a red algae, and this is a brown algae. Named in the past, of course, because of the way they look. These are also algae. So this is a volvox. It is a simpler form of algae, but it is multicellular. And then algae go all the way down to these single cell types, which are the topic of interest. And these are the types of cells that we actually are using as our biofuel feedstock. I wanted to mention here, so if we're going to go down this rabbit hole, we have to understand, if we're trying to understand where algae actually come from, there's a few terms we have to learn here. So algae are eukaryotes. Can I get a show of hands if anybody in the room knows what a eukaryote is? Couple. Okay, so the biologists in the room know what a eukaryote is, right? So a eukaryote is a particular type of cell, okay? And if we look at the tree of life, this is a very simplified version of all of the organisms on the planet. And what we know, based on the genomic and DNA content of these organisms, that there are 3 different types of cells. We have bacteria, we have archaea, and we have eukarya, or eukaryotes, okay? So you and I fall in this portion of the tree, along with algae, plants, all animals, fungi, etc. And then we have bacteria up here and archaea over here. But if we focus in on a particular portion of the bacterial part of the tree of life, we see 2 interesting components, right? We see a chloroplast and we see a mitochondria. So if you study evolutionary history, what you know is that the chloroplast and the mitochondria, right, are descendants of very primitive bacteria. And how this occurred and how algae evolved, it occurred in several steps. So there were 2 what we call primary endosymbiotic events. So an endosymbiotic event basically is one cell decides to engulf another cell and to form a new species. So billions of years ago, there was some type of a proform, or an early form of this eukaryotic cell. And at one time, it engulfed what we know today as an alpha proteobacteria. So we know it's an alpha proteobacteria because even in modern cell types, we still have DNA in our cells that indicate that this is an alpha proteobacteria. So this process occurred, like I said, many years ago, and it formed what we know today as a modern eukaryotic cell. And then many, many generations later, that cell has evolved to form humans and plants, or humans and fungi and animals. Now, and as far as we know, this remnant, or this particular remnant of the alpha proteobacteria, is what we know today as the mitochondria. Now, this only occurred once in evolutionary history as far as we know. But there was a second event where this proform, or this proeukaryotic cell, it actually happened again. But in this time, it didn't engulf a proteobacteria, it engulfed the cyanobacteria. So cyanobacteria are a little bit different in that they are actually photosynthetic as well. And so you get a secondary engulfment, and that ancestor of what we know today as the chloroplast was actually a cyanobacteria. And then once again, many, many generations and billions of years later, we get what today we have as modern plants and algae. From that point, I mean, it's important to stress here, what we're talking about is these are the primary events. This didn't just happen once. Well, the engulfment of bacteria into a eukaryotic type cell only happened twice. But these eukaryotic cells started engulfing each other over the billions of years. And what led to this massive diversity that we currently see today. And so when we go back and look at the common names for algae, we've named them red, green, brown, yellow. And it really has to do with the pigmentation and how they look to us physically. But in actuality, the lineages look a lot different if you just look at the evolutionary phylogeny. So most of those organisms are all, if you see a red algae or green algae, brown, those are what we call true algae. But once in a while, you'll see this term called blue-green algae. And these are really not algae at all. What these really are is they are cyanobacteria that have been around for a long time, and they continue to be cyanobacteria. Now, algae are grown for many different reasons and many different purposes. We currently use them in aquaculture. We feed them to fish. We use them in nutraceuticals as nutrient sources. So I actually bought a bottle of juice from Smith's a couple hours ago. And if you turn the bottle over, I need a volunteer. Volunteer. I'm not going to ask you to drink it. Just read. So read the top two ingredients on that bottle.
Chlorella and spirilla.
Yes.
Spirulina,
Chlorella and spirulina. Okay. So those are actually quote unquote algae. So chlorella is a true algae, and spirulina is not really an algae. It's actually cyanobacteria. So there's precedence, in industry to use these materials for human consumption, but we're trying to use them in a different way, right? We're trying to use them and grow them for biofuels, which presents some challenges, and I'll get into those in a little bit. All right, so this is the eukaryotic tree of life. So we simplified it. We're not going to talk about the bacteria and the archaea. We're just going to focus in on the eukaryotes. And it really is the same version of this, just represented a little bit differently. And what I've done is just to orient you, I've highlighted with stars different types of algae that are currently candidates for biofuels that we're going to use to convert into biofuels. And as you'll see here, humans, are located on this portion of the tree, and plants are located over in this portion of the tree. So in academic circles, they study only a small fraction of algae. And in particular, in the past, they've mostly focused on this, this lineage of green algae over here. Now, some of those green algae are actually pretty good candidates for biofuel production. But what these stars are supposed to help you indicate is that there's a large diversity of algae out there, and they all have specialized and individual functions. And so we and others in the field are trying to understand the differences in how these different types of algae grow, and we're actually trying to harness the abilities of all these algae, depending on conditions, to optimize production. Well, so how does this really work? If we first consider oil, right? So we're taking oil, fossil fuels, we're pumping it out of the ground. It goes through an extraction and conversion process. It gets refined, and it gets converted into jet fuel, diesel, and gasoline. So in the case of algae, all we're trying to do really is, change the front end of that system. So we can't pump it out of the ground. We have to actually grow them. So that's really the only difference. We're trying to harvest, take the energy from sunlight, trying to recapture some of the CO2 from emissions or in the atmosphere and convert that energy and carbon dioxide in these open production systems into algae. And we're interested in all different components of the algae. So we're interested in the total biomass, how many cells can we actually grow. We're interested in the fat content and the carbohydrate content. And the different types of lipids and fats that the algae have will influence what type of biofuel you get in the end. So some people are really interested in making jet fuel. Others want to make gasoline. Others want to make diesel. So if you're interested in one particular downstream product, you're probably going to select and grow different types of algae. So what makes algae special? Why are they good candidates for biofuels? Well, the first reason is they can store a lot of carbon. So what this image is, this is a Nanochloropsis algae, and the image at the top represents very blown up images of these algae under normal growth conditions. So they're presented with plenty of food, they're growing happily, and we can easily see, we were talking about the chloroplast, here's the mitochondria in these cells, and you'll see a few, what we're calling here is oil bodies or lipid bodies inside the cell. And then if you starve the cell, you remove the nitrogen from the medium or the content, the cells basically cannot grow. But they still want to be able to store carbon when nutrients become available again. So they're basically hedging themselves. They're saying, okay, if I can't grow, let me just start to store some of that carbon material. And so you starve them for nitrogen, and lo and behold, what they start doing is filling up their cells with oil. And so it's this component that actually we're using to convert into biofuels. Okay? So we're utilizing what exists in nature. It's a natural event that occurs. We're just inducing it in an artificial setting, in a production setting. The second reason algae are good candidates is that they really can grow and thrive in a diverse amount of habitats. So you can find algal blooms in Arctic seawater. You can find them up in alpine freshwater lakes. Algae grow this is a picture of some algae growing in Yellowstone National Park in some very acidic thermal hot springs. And algae definitely grow in ditches. There's lots of runoff in farm environments, and all those nutrients are in there. It just selects for algae, and they can grow and bloom quite nicely. And then here's some images of algae growing inside coral. I don't know if you actually know that, but actually the color you see in coral is actually coming from a symbiotic event between the coral and the algae. And when you get bleaching of the coral and they all turn white, what's happening is all the algae are dying, and that's why you're left with the white calcified material. So this is good news for us, right? So no matter what type of climate you're growing in, if you want to grow algae in the upper parts of the United States, or you want to grow them in the Southwest here, there are particular types of algae that can grow in different regions. So the other thing that does this really make sense in terms of land mass, right? Because we have to actually use land to grow these algae. And let's see, how much land does that actually take? So this estimate was completed by Anthony Marchese at Colorado State University. And what he uses as a standard, or as a benchmark, is 21 billion gallons per year of advanced biofuels. So in 2007, Congress passed a bill which sets our renewable fuel standard at approximately 10% of U.S. consumption of 10% of U.S. consumption of liquid fuels needs to come from a renewable fuel source. So if we were to generate that 21 billion gallons per year from, let's just say, soy diesel, which is currently being utilized across the United States, that would require a landmass approximately the size of Alaska. So it's not really practical to think that soy diesel in and of itself is going to be a long-term solution. But what's the case for algae? So we can, with a landmass about the size of Connecticut, we can produce that same amount of oil. And this is much more reasonable, considering that we can also grow these algae on non-arable lands. We don't have to compete for agricultural lands. We can grow them, for example, in the desert, if there is a source for water. And then there's a couple other advantages I'd just like to quickly highlight. Algae grow very fast. In fact, they grow 10 times faster. We can generate biomass from algae 10 times faster than land plants. As I mentioned, they are non-competitive with agriculture, so we can use non-arable lands to grow the algae on. We can also grow them in all different types of water. We don't have to compete for fresh water sources. So we can use wastewater streams. We can use saline and brackish water. There are some companies in New Mexico that are actually looking at using produced waters, so wastewaters from the oil and gas industry itself, to grow the algae in. And I think the biggest benefit, one of the biggest benefits, is that it plugs into our existing infrastructure. So we're continuing to convert these algae and all of the materials inside the algae into existing fuels. And so it'll fit into the downstream refining processes that are already in place. So let's bring it closer to home. So why do we really want to cultivate algae in New Mexico? This image is a picture of a test facility that Sapphire Energy currently has in southern New Mexico in Columbus. And this is just to give you an idea of scale, this is a 100-acre build-out of a pilot program. And what you'll see here, it represents nicely, there's lots of flat land. So we grow the algae in very, very shallow ponds, so 10 centimeters. So we do that because we want the light to penetrate the entire pond depth. We don't want a large reservoir body of water where the light isn't penetrating. We're not going to get any growth. So that means we have to spread these ponds out horizontally across large land masses. And the other good thing about New Mexico is relative to the other places in the United States, we have a larger growing season. So especially in southern New Mexico, they don't have as many freezing nights. We have sunshine and more constant temperatures. And then, of course, we have access to brackish water. So down in southern New Mexico, if you go into the aquifers, it's saline water. It has a lot of salt in it. So we can't use it for drinking water, but we can actually grow algae in it. So I told you all the good things, but now we have to balance this out and talk about the bad things, or what's limiting us in the field. What are the current challenges right now that we're facing? And the biggest thing is it costs too much. It's really hard to compete with fossil fuels, which are just pumping out of the ground, versus something you actually have to grow from scratch. That takes time, it takes energy, it takes infrastructure, and that's really the only thing that's impeding it. It's not that we can't do the downstream conversions and make the fuels. That's kind of already been taken care of. It really is, we need to bring the costs down. So I was a part of a large consortia called the National Alliance for Advanced Biofuels and Bioproducts, and this particular consortia was run out of Los Alamos National Laboratory. And as a part of that project, the techno-economic analysis team, led by James Richardson and Megan Downs at Texas A&M and New Mexico State, they took some of the improvements we were seeing in all of our studies and created several different scenarios. So if we look at our baseline here, and we say, okay, if we have this is current capital expenditures and operation expenditures, what that leads to is a really large range of fuel cost in the end, $15 to $200 a gallon. All right, so this is really not feasible. No one's going to pay $200 a gallon for a gallon of gasoline or a gallon of diesel. But if they started implementing these improvements with various different improvements at different stages of the process, in the harvesting, in the extraction of the oils out of the algae, in the cultivation, and in the biology, we see these numbers start to decrease. So if we were actually able to implement all of the different technologies we've actually developed at a small scale and implemented them together, we start to get actually into that range where we can be cost competitive with fossil fuels. So, but therein lies the challenge, right? We can do some of these technology improvements in a flask in the laboratory, but that doesn't translate to growing them at large scale outside in southern New Mexico. So really the goal currently now, this NAB program of the National Alliance for Advanced Biofuels and Bioproducts kind of set the baseline of the feasibility. Now what we're trying to do is moving on, and the Department of Energy has funded actually scale-up studies. So now we've demonstrated this in the lab. Let's see how we can actually grow this outside and take on the next set of challenges. All right, so I covered the too expensive part, but the other part that's really kind of bad is that really it's just too dry in New Mexico. So I mentioned that these ponds are 10 centimeters deep. All right, so when we think about evaporation, that means that a lot of that water is going to evaporate really quickly. What happens then is that it becomes, you turn a saline environment into a hypersaline environment, and of course, a lot of algae don't really like that. And so you have a lot of evaporative losses. So the test pilot ponds that I showed you from Sapphire were all open systems, right? So they're exposed to the environment, you get a lot of evaporative losses. So Pete Lammers at New Mexico State University started to grow these in southern New Mexico in enclosed systems. Okay, well, so it kind of takes care of our evaporative loss problems. You still get water being evaporated, but we can recapture that if you enclose the system. And what you'll notice here is that these systems in particular, so what you can see inside here is a mini raceway. It's just a circle of algae that are being floated around inside, covered by a cylindrical tube of plastic. Okay. And here's a little more an advanced version of this and a little bit longer. This is really as advanced as the systems are going to be. Because like I said, we're trying to compete with a commodity. We're trying to compete with fossil fuels, and we need to keep the infrastructure as cheap as possible. But this presents a problem. As soon as you do this, what happens? It heats up. Yeah, so it's like a little greenhouse. So we've taken care of the evaporative loss problem, but now what happens is we've caused the temperatures in this environment to increase. So this represents, annual cycles of temperature inside similar types of what we call photobioreactors, or a production system. And the maximum temperatures we're seeing in those production systems are 55 degrees going down to 25. So these lines, these lower and upper boundaries, are kind of the limits of growth for some of the early algal strains that we were playing with. So they don't like to grow. They can survive at this temperature, but they don't actually grow. And when you factor in these lower and higher regions, what this means is loss in productivity. And half of your year is basically you're operating your production facility under suboptimal conditions. And what this figure demonstrates is really, we decided, well, let's see how well this particular type of algae can grow at a range of temperatures. So what the y-axis indicates is maximum specific growth rate. So how fast are these cells dividing and growing? And then looking at that growth rate at different temperatures. So what we'll see is that Chlorella sorokiniana, one of our putative production strains, actually grows quite well until you hit about 35 and then rapidly declines. And so what we realize is that while this strain may grow very well in the winter in southern New Mexico, it's not going to survive and give us any kind of productivity in the summertime. So we needed a new strain. So we turned back to the environment. Well, most of the algae that we've played with so far have been pretty intemperate regions, right? So they're in fresh water, then in the ocean. You don't get a lot of change in temperature in ocean water, in the open ocean. But there are environments that actually do select for algae that like to grow in hot environments. And this is the type of environment that most closely mimics our production system. So even though nature has done its job and these algae have occupied virtually every niche of the planet, we're introducing yet a new type of environment, this production system, that we're trying to optimize these algae to grow in. And so we're starting with, let's just say the closest thing we have to what may potentially grow in this type of environment. So let's go back to our evolutionary tree. So this particular type of algae is a red algae. It's a Galdiaria. And this algae it grows in Yellowstone National Park, and Pete Lammers decided to grow it in southern New Mexico and see if we could actually get it to thrive. And what's unique about Galdiaria, if we consider the temperature limits of pH in life, that this thing grows at near pH of 0 or less than 1. So not many things can grow at pH 1. This is a good thing for us because it helps select and decrease the contamination in the system, and it optimizes production for the strain of interest. And what we see is that these are also mesophilic, and they can grow up to 55 degrees, just about the same temperature that we're looking for. So last summer, Dr. Lammers got a hold of a culture strain of Galdiaria sulphuraria and decided to grow it in one of their closed raceway systems. And this is what's considered a typical 3-week production cycle. We start the cultures, we watch them grow for 3 weeks, and then we harvest. The red lines indicate the fluctuation in temperature over that 3 weeks. And what we see is that the low temperatures in the summertime down in Las Cruces are around 25 degrees, and then inside they max out over at 55. And what this black line indicates is the level of growth. And over time, the algae really seem to thrive at that 55 degrees. So over the last year, we've added a new strain of algae that we're considering to use as a biofuel production strain. But the problem with this particular algae is that while it handles the high temperature, when you try and harvest the material out for biofuels as a feedstock, you only get about half as much feedstock as you do from growing it with chlorella. So we kind of have a problem here. We have chlorella that grows really well at ambient, normal mesophile temperatures, and it grows very well. And then we have another strain that doesn't grow quite as fast, or we can't extract as much lipids from it, and the other strain. So really what we'd like is to, take the best of each algae and put all of those traits together. So we started, Amanda Berry and Dr. Sayer at Los Alamos National Laboratory and the New Mexico Consortium started a directed thermal evolution study. So they decided to take one of our best biofuel candidates, Chlorella serocaniana, and see if we could adapt it to higher temperatures. And what we're doing is taking advantage of normal evolutionary processes. So others in the field, or in algal biology and evolution, have been studying the mutation rates of algae. And these particular some types, a related algal strain, on average, generates 100,000 mutations every day. So if we force these particular algae to grow under higher temperatures, under different increments, and we subject them to this type of selection, can we actually select for mutations that improve its growth at higher temperatures? So this particular project was started last fall, and Amanda and Dr. Sayer started growing it at 39 degrees. And what you'll see down here, these are what we call photobioreactors, and these mimic growth inside our production systems. So the depth here can simulate growth up to 20 centimeters. So we grow them at the same depth. The light is coming from the top, and these are all hooked up in serial, and you'll see these tubes coming across the different photobioreactors. So this one is set up at 39, this one is set up at 41, 43, and so on, and we have these 2-degree increments. And the algae are allowed to pass between each one of these different types of reactors. And over time, what we've seen, after a couple of months, we started to see algal growth at 43 degrees. And then after 3 months of growth, we started to see growth at 45. So to make a long story short, what's happened now currently is that we're starting to see them max out. We haven't seen much improvement beyond 47 to 49. Sometimes they'll show up at 47 and 49 and then they'll go away. So we haven't seen a stable mutation that gives us good growth at these higher temperatures. So the next tactic really is to do what we call chemical mutagenesis. So in this particular sense, we're just letting the cells evolve and mutate naturally. But if you add a chemical mutagen in the laboratory, you can actually get them to evolve faster. And those studies are ongoing. So we're trying to increase the rate at which they're creating mutations in the cells so we can select for algae that actually grow at higher temperatures. Okay. The last case study I want to talk about is adaptive evolution of a green algae called Picochlorum. And this work is led by Scott Torrey and Tarika Dale in the Bioscience Division. So what these images represent is the lipid content of the cells are all stained. So the bright green means that there's a lot of lipids inside those cells. Whereas in these other types of cells, there's a lot less. There's a lot less, the lipid content is lower. And this is actually very normal. So in a culture, despite the fact that you start with a single cell and you let it grow and propagate in that environment, you get this heterogeneous mix of different types of algae with different amounts of lipids. And if you analyze all of the different types of algae inside that culture, what you'll see is there's a distribution of lipid content. And we run these cells through a flow cytometer and actually analyze every single cell. And here's the distribution of those cells. So you have this average, but within that average, we actually have high lipid producers and low lipid producers. So Terica decided, well, what if we take only those high lipid producers and we throw away the rest? Okay? So we're going to run these through our fancy flow cytometer and sort out and keep only the ones that are high lipid producers and then grow those up. And then we do that again. We go through, run them through the flow cytometer, select only the highest lipid producers, and we grow them again. So we're constantly selecting for a high lipid producing phenotype. And so we did this 4 times, and the next few slides are going to refer to a couple different types of organisms, the high 4, which means it went through 4 rounds of selection, and then also we're always comparing it to our wild type or the native population, the heterogeneous population that we started with. Okay, a lot of graphs here. So once again, the graph on the right here is indicating nitrate content. So this is our nitrogen source. So if you think back to the very beginning, when we starve these algae for nitrogen, they actually start induce lipid formation. So these lines here represent the nitrogen consumption in these cultures over time within the first 4 days, and what you'll see is they consume all the nitrogen. About that same time, what you see is an increase in the lipid content. So this is the lipid content in the high 4, and this is the lipid content in the wild type. And what we'll see is that those algae actually produce a lot more lipids, almost twice the amount of lipids that we've seen in our wild type culture. So we're actually, just by doing this sorting and selecting the highest lipid producers, we can double the amount of lipids that we're producing, which directly relates to a halving of the cost of production downstream. And this is done with very little loss in biomass. So we get a doubling of lipids, but we only lose a little bit of the biomass. So the basic scientists in us really want to know is like, why is this occurring and how is this happening? And so my job at the laboratory is to study the genomes and transcriptomes of algae. So what we did is we harvested the DNA from that high 4 selected population, and we harvested the DNA from the wild type, or from that native population, and compared the genomic content. And we looked at every single base in the genome. So this genome is 15 million bases, and we compared each position of that genome and looked for differences. So this is the list, the comprehensive list of all the mutations that we saw in that high 4 genome. So many of these are at the repetitive regions at the end of what we call a contig. That's just an assembly of a long piece of DNA. And these repetitive regions exist in all of eukaryotic cells. And so it's really hard to determine really what that really means, if there's any function whatsoever. But what we noticed is that there was a mutation in a regulatory gene. So it's a particular type of a gene in a system that controls the expression of thousands, potentially thousands of genes in this particular type of algae. And so it's a very, very important gene. And this particular single base mutation may actually account for that phenotype with the high lipid producing strain. So we not only looked at the genomes, but we also looked at the transcriptomes. So back to our biology 101, the genes in our system, they encode all the information to generate all the things we have in our cell. And so that information gets translated into transcript first before it gets converted into proteins. So we looked at the entire transcript of the genes of this particular high 4 and compared it to wild type. And what we see is that a lot of these genes are expressed very differently. And even over time, we see a lot of genes that are overexpressed and a lot of genes that are underexpressed. And if you compare it, if you look particularly at the genes that are involved in synthesizing all of these lipids, what we saw is that all of these genes are expressed much faster in our high 4 population compared to our wild type. Now, we don't really know why yet. So the genomic mutation gives us an indicator that potentially what our current working hypothesis is that these particular algae are sensing a nitrogen limitation earlier than the wild type. And so they basically think they're starved when they're not. And so they start storing away all the lipids earlier than the heterogeneous population. And I should say that this subpopulation has been very stable. We've continued to grow it in the laboratory, and it still maintains this particular phenotype. And so what we're doing now is basically following up on this and trying to identify a mechanism. We're doing knockout studies, what we call. We actually took this particular organism, we substituted this particular base in its genome, and we're going to see if we can reproduce that phenotype. Okay, I'm going to stop and open the floor for questions, but before I do that, I wanted to acknowledge all of my co-workers and co-authors on this work. So the Piccochlorum adaptive evolution work is led, like I said, by Scott Torrey and Tarika Dale, with assistance on the biochemistry from Cliff Unkefer, and these are all staff scientists in the bioscience division. The Galdiaria work with the red algae and the chlorella growth studies are done primarily by Pete Lammers and Wayne Van Voorhees at New Mexico State University. And one particular study was completed by Michael Huseman at Pacific Northwest National Laboratory. The thermal evolution studies of chlorella, this project is run by Richard Sayer and Amanda Berry. And when we get to that particular point to study the genomes, we're also interested in the genomes of these thermal evolved studies, then that's when I come in and do the work. But they pretty much are running this project. And then I have to thank my current and former students, because I wouldn't have time to put talks together if there wasn't actually somebody working in the lab. Jennifer Kwan, who's currently at the NIH, and Tumpa, who is a student at UNM now, and Juliet Ohan, who's at the laboratory currently. And funding for this work comes from two sources. So LANL itself pumps money into this to continue to develop this research. We get research dollars from laboratory-directed research and development funds. And the Energy Efficiency and Renewable Energy Office funds the majority of this work. And that comes from the Department of Energy. Thank you.
Thank you, Tom. Wow, that was absolutely fascinating. Really great stuff, very exciting, like hard to imagine the future, but maybe we can. Who has questions for Shawn? Who might have a question? Here's one.
Did the oil we get today come from algae, a part of it?
Yes. Yeah, so the coal beds we have deep down, we know that that's basically compacted plant material. There are particular types of algae, I didn't actually talk about them here, that have very similar lipid profiles of our current crude oil. And they have basically demonstrated now that some types of algae that crude oil is actually deposition of old ancient algae. So the ones we're growing right now may not exactly be the same type of algae, but because that they are ancestors of those algae, they do produce similar types of, I would say related lipid profiles that we can actually just change the refining a bit to select for the right carbon molecules to convert into transportation fuel.
There's another question over here.
So coal is compressed algae?
Compressed plants.
Photosynthesis.
Yeah.
Got a couple questions. One is, where is this photo taken?
This photo, this is a company called Cyanotech, and they grow spirulina. Okay, so the cyanobacteria, they don't actually grow algae, they grow cyanobacteria. And this is their production facility in Hawaii. And they harvest the cyanobacteria for different nutrients, nutraceuticals, kind of like the same stuff I was drinking in that juice drink. But you can buy the little pills. And then a lot of algae are actually grown for beta carotene. You can consume a lot of the, in your vitamins, that beta carotene actually comes from algal production facilities.
The second question was the enriched lipid algae that you're working on. What do you see as a future possibility for that?
How so? You can be a little more specific?
Well, what research are you going to do in the next year, I guess?
Okay. So I kind of alluded to this. So it really is trying to demonstrate at scale what we've seen in the laboratory. So we can, if you work all the numbers and if you translate the densities and the type of lipid production that we're seeing in the lab scale, and you extrapolate out to large hectare farms, where actually can be very cost competitive, with fossil fuels or with crude oil. But making that transition is very difficult, and it's a challenge. So it's not just an easy jump to grow it at scale. So we're really focused on understanding how we need to grow the cultures, at these intermediate scales so we can expand the system. The second thing that we're working on is a lot of genetic engineering. So Dr. Sayer at the Mexico Consortium has several different strategies to re-engineer or basically combine interesting traits in a focused way, in a targeted way, to further improve the production, the growth, and the lipid content of the algae.
So what dollar per gallon do you say is current, and what?
So
would you expect
Right.
in
So
by-
right now, it's a little high. It depends on what technology you talk about and which technological improvement you actually implement. So if we were able to implement at scale all the technological advances that were created over the last 3 years, we would be cost competitive with fossil fuels. But the big assumption is, can you do it at scale? Oh, they want to, like.
Okay, I had a couple questions.
Sure.
First of all, when you talk about the 10-inch pool depths,
Yeah.
so algae grows within that full depth, and then you skim it off in harvesting every three weeks, 3 months. How often?
So the production cycles, it depends on the company you work with or what their thoughts are. Some run what you would consider a continuous culture. So you would only take a fraction of that material off every time, and so the algae just continue to grow in that system, and you're always just siphoning off a little bit.
So you don't have to reseed it.
So you don't have to reseed it,
Okay.
right? Some people like to do an inoculate every single time, so it's almost like a batch system. You inoculate, you grow it up, you harvest everything, and then you start over.
Okay.
So there are advantages in that way because you're always, the longer you leave the cultures running, the more chances are you're going to contaminate with predators or other types of algae or other creatures you don't want, in your crude. And so that there's advantages, both ways.
Okay, because with the batch type system, is there any thoughts on running like a summer batch and a winter batch that are more specifically adapted to
Yes.
the temperature, similar to
So
agriculture?
it's a crop rotation.
Okay.
So that's really the current working strategy is if we can't improve, if we can't make Galdiaria grow better in the winter or we can't make chlorella grow better in the summer, then yes, we'd have to you'd switch out the strains. You grow Galdiaria when it's hot in the summer, and then you grow chlorella when it's cooler. And then overall, you average out that biomass, that productivity you're going to actually improve. So that is definitely a strategy that we're considering.
Okay, and then one last question. I've forgotten a lot of the differences with lipids versus carbohydrates, but
Yeah.
I guess in my understanding, the photosynthesis creates the simple sugars, carbohydrates, but then the cell converts that to fats.
Yes.
So the conventional fossil fuels that we usually use are long chain carbons.
Yeah.
How long are these fats, and does that take a lot of energy for the cell to then switch from sugars to fats in kind of a starvation mode?
So
Can you describe that at all?
yeah. Very complex answer. So depending on what type of algae you're growing, different algae make different types of starches, carbohydrates, and they make different types of lipids. Some prefer to store their carbon as starch. Others like to dump it all into lipids. And then within that lipid pool, different algae make different types of lipids. So a particular strain of algae called Nanochloropsis is actually grown right now, and we harvest it for very, very long carbon chain molecules, EHA and DHA, what ends up in baby formula. It's these long fatty acid molecules, these healthy fats that the algae make. So they make very long ones, but then they also can make very, very short, they make shorter triacylglycerides around carbon chains 14 to 18. So there's really no simple answer. Depends on what type of algae, depends how you grow it. Some of the algae that primarily convert a lot of that CO2 into starch, we found out how to actually make them switch over and convert it all into lipids. So there's all different ways, all different things you could consider.
Okay, but once it's a lipid, you can burn it.
Oh, yeah. So the lipids in and of themselves, right. So the lipids make a better starting material to convert it into biodiesel. So there's a carbon backbone for these triacylglycerides. You knock off that carbon backbone and you have these long carbon chains which are readily convertible into diesel. If you're interested, and then there's another person in the laboratory who's really interested in converting all the starches directly into gasoline. So he's figured out a way how to make that conversion actually quite easily. So.
How much algae do you need to make a gallon of gasoline?
Let's see. So the algae so you remove most of the water content of the algae, and it gets compacted into kind of a paste. I should have put a couple slides in there. And that's what becomes our biocrude. And then what we do is we put that through a process called hydrothermal liquefaction. That's just a fancy term for a big pressure cooker. So you heat it up to high temperature and pressure, and what it does is it simplifies all the complex molecules in the system, and you get out different fractionations. And it becomes your, it moves it from crude into kind of a pre-refined product. And you get a water phase, and you get an oil phase, and an intermediate phase for all of the proteins and stuff that didn't break down during that hydrothermal liquefaction process. So I've never, depending on how efficient how much lipids are in your initial system and what particular fuel you're after, that varies. So I can't really, I can give you a large ballpark range. I can't tell you.
For, let's say we had like that Sapphire
Yeah.
plant, how much area of that plant might be required, do you think, roughly?
Yeah.
Ballpark, to make a gallon of gasoline?
I might provide you with a better number.
It's just a ballpark idea, that's all.
Yeah, I hesitate to even say. I don't want to be wrong.
Okay.
It really is dependent on the system, what fuel you're after. Yeah, I can't.
Who else had
There was a
question
question in the front here. Yeah.
Sir. Well, it won't pick it up on
Okay.
our
All right.
recording.
Okay,
We don't
that's fine.
have you do it.
With your enclosed systems, do you need to replace the carbon dioxide that gets used up? And then the second question is, what do you consider at scale? How many acres of algae, would you consider in order to get down to that low price for fuel?
Yeah. So if you recall one of the earlier slides that I had, I'll answer the second question first. Let's see. Here. So kind of the gold standard here is to, use algae to meet this renewable fuel standard, so 10% of the consumption of U.S. fuel in the United States. And if we were to grow algae and convert all that algae into biodiesel to meet that 21 billion gallons, that's the land mass it approximately would take currently. That easily could fit within, the regions of southern New Mexico that we're considering to grow these algae in. So it's in that sense, it's very feasible. Now, your first question.
Would you need to replace the carbon dioxide that gets consumed in an enclosed system?
Yeah, so it's not completely enclosed. So we can pump carbon dioxide into that system. It's just not open to the air. And we can, through a filter, you can add more air into that environment. And that is done. You can't cut them off completely. Yeah.
Right now, the federal government subsidizes a lot of corn growing for gasoline additions. Area-wise and percentage-wise, do you know how the corn production compares to algae production?
Yeah. So there's two things to consider. If you're using the kernel to make ethanol, or if you're using the stock or the cellulosic material in that plant to convert it into ethanol, for example. So in the case of using the kernel, your land mass is about the same here. Okay? It'll take that much land mass to make that much fuel. When you consider cellulosic sources, that square gets compacted to about a third. So if you notice that's still much larger than our estimates for algae. So it is an improvement by moving away from the kernel and just using the stock. You heard this term cellulosic ethanol production. But it boils down to growth rate of plants. So algae grow 10 times faster than plants do, and it takes a lot longer time to grow that material. And so you're always going to be at a disadvantage just based on growth rates.
But our politicians can't buy votes with algae.
What's that?
So the politicians can't buy votes with algae.
No, they sure can't. You know, so you're fighting against so the Department of Energy has pumped a lot of money over the last 20 years into biofuels from ethanol and cellulosic ethanol sources. And so they want to see a return on that investment. So we are kind of at the we're just kind of getting started again. You know, they've been pumping money in for the last, 3 to 5 years. And so if they would fund, this is my biased opinion, not of the laboratory or anybody else, if they would fund algal research at the rate that they're funding cellulosic research, I think this is very doable in, I'm going to say 20 years. We'd be ready to roll.
Shawn, who else is doing research on this, either here in the country or around the world? And is it a tremendously competitive research environment to get to the answers that you're trying to get to?
So the United States is actually lagging behind in dollar for dollar research towards algal biofuels. So China and Korea, Australia, some of the European countries are pumping a lot more money into algal biofuels research than the United States is. So yeah, everybody around the world there's a lot of researchers that are interested in it. There's no silver bullet yet. And I guess my take on it is we need to be focused on growing algae in the region. And those algae are going to grow the best in New Mexico, and we're going to select for strains and adopt and optimize those strains for growth here. Now that they may not grow the same in Australia or Korea. And so I think they're focused on their own industry and their own local climates, and my thought is you're going to have regional solutions. And so I don't really feel like there's a lot of competition outside of what's happening here, So.
Can this process run away and catch fire?
Catch fire?
Yeah.
No. No more so than, once you get to the refining state, then you're in the same boat as, fossil fuel, refining of crude oil. But in the ponds, no, you're not at risk of flames, no. Not going to start up, combust.
So what's the downside in terms of pollutants that might be produced by this? Anything?
So in order to qualify as an advanced biofuels, it means you need to reduce your carbon emissions by 50% over current production of fossil fuel sources. So based on current estimates, algae meet that criteria. So we're at least going to be reducing carbon emissions by half. And what we're trying to, you'll never be net carbon neutral, but you're trying to reduce the amount of carbon that you basically waste in the process.
So I was just wondering whether that process of selecting for the perfect strain that would grow in that particular part of New Mexico can be simplified by just like doing this cycle several times in exactly that area, and then in the end you're gonna basically and then seed stuff that grows easily, and then seed the next crop with whatever grows easily, and go on just naturally in such a way that you reproduce exactly the condition that you want. Because it seems to me that like what has been done so far is like compartmentalized. So there's like maybe Taraka is doing UV irradiation to induce mutation. Dr. Sayer is doing like only temperature. Whereas over there you have temperature, you have salinity, you have contaminants. You have those covers that inhibit, allows a certain amount of light through. So I was just wondering whether it wouldn't be easier to just like do it naturally like that.
Yeah.
So have you, for example, like compared the genome of the starting population to the genome of the population that has evolved over time,
Yeah.
for example?
So I think you're thinking around the right lines, and I think the exact same way. A lot of research dollars have been wasted with playing with various algal strains in the laboratory that don't pan out once you move them into a production facility. But to me, though, the resources aren't available to do the types of studies you're indicating at scale outside. And so to me, there's always going to be a balance between what's happening, the small scale studies in the laboratory, and then the larger, the studies outside, at scale or at some type of a larger scale in a more real world setting. But so we are actually focused on moving algae both directions. So we're growing the algae down in New Mexico right now. We're trying to get a handle on how it grows and develop a stable population outdoors, and then understand what that population looks like, and then use that particular strain and move it back inside, and then try and involve it with more targeted improvement methods. So we're kind of working both ways, and our collaborators at Michigan State University have done a really good job trying to simulate what's happening in those outdoor production facilities. So they built this particular those photobioreactors that we're using in the laboratory to mimic as closely as possible what's happening outside. I mean, with some limitations. And so our focus is always on, well, how do we make sure that we get the most bang for our buck with our lab work? But I think it's really a combination of the two that's going to, make us successful in the end.
And with that, thank you very much, Shawn. That was really
Thank you.
interesting.
Thank you.
you.